The big question is, why are multiplicative updates so prevalent in nature? Maybe they're really not, but I just see it that way. Are additive updates used? And are the matrix version, that's the final question, of the multiplicative updates used? That's how we'll conclude. Okay? Is it clear? Good. So here is the classical application of a multiplicative update. This was done first in the weighted majority type setting. So you have a set of experts that predict something on a trial-by-trial basis, let's say the weather. The master algorithm keeps track of the experts by, it keeps one weight per expert. That's the belief that the ith expert is the good expert, right? So wi is sort of the belief in the ith expert at trial t. And then your update is something like this. The new weight is the old weight times a factor. Same thing as in boosting, right? Distribution on the example in AdaBoost was new weight on the example is old weight on the example times e to the minus eta loss, right? So here it is, new weight is old weight times e to the minus eta loss I, normalization, because I keep a probability vector. My uncertainty vector is a probability vector. Later on, I'll generalize that to matrix. Eta is a learning rate. Z is a normalizer. Bayes update is a special case, very important, when your loss is the log likelihood. Then this would be the data log, minus log of the data likelihood. Eta is 1. And then it becomes the prior times the data likelihood over normalization posterior. Yeah? So it's just a special case. Special case of multiplicative update is Bayes. Yes?
Why does it make sense to have a probability distribution over the experts?
Because it's your uncertainty vector over the expert.
But they're not mutually exclusive.
They're not, but you model them. In the vector case, you model them as sort of mutually exclusive. Yeah? In Bayes, you have m different models, and you keep an uncertainty vector over the Bayes models. Yeah? This is your prior, and then your posterior, and you update the posterior, and so forth. Yeah, that's the same philosophy. It's just a different loss. Big picture, just a different loss. Okay? So Bayes update is a special case. So it seems to be that this is sort of natural when you have concentrations, these kind of updates. Yeah? Because you have only positive weights. When you have a concentration, you don't have a negative concentration. So this is my gut level answer to why multiplicative updates are used and gradient descent is not. Because in gradient descent, you subtract the gradient, you would get negative numbers. How are you going to do that with concentrations? You have to do some kind of a trick to do that. But if you have concentrations of chemicals, ions, or whatever it is, you naturally get into these updates. The number of ions goes down. The number of ions goes up, maybe by multiplicative factors. If you do additive factors, then you have to do something around 0. You can't go negative. But this might be too simplistic. Okay? I'm not a chemist nor a biologist. I just have fun. Okay. So next, I'm going to do an implementation of Bayes in the tube. And here's my tube. Right? You can use a beaker. I usually travel with a beaker. And you have your DNA solution. And our computation is going to be poured back and forth. I'm serious. That's the computation. And you can't do it with a computer, because computers are too slow. You will see. No chance. The blessing of the multiplicative update, I claim, is the speed. The curse is the loss of variety. The loss of variety. And there's going to be deep philosophical questions, as you will see. We're going to give 3 machine learning methods for preventing the curse and discuss related methods used in biology for preventing the curse. Now, of course, nature, the evolution is not directed, but only those evolutionary systems that ameliorate the curse of the multiplicative update will survive. That's what I claim. The others don't. That's why we have predators, and that's why we have these, we have this complicated thing. You have a caterpillar, and then you have a wasp that lays eggs in the caterpillar, and then you have another wasp that parasites on the parasitic wasp. This is all very important, I claim, to prevent the curse of the multiplicative update. Totally interesting. Sicknesses, very important. Okay? Good. So, in vitro selection. I'm going to do now these updates and the learning algorithms and the base algorithms in the tube. Right? You have a tube with a liquid in it, and there's RNA strands, and you're going to compute by pouring the tube. So here's my tube, blum. I want to find an RNA strand that attaches to a protein. Why am I so fascinated by attaching to a protein? If you want to design any kind of a drug, first thing you have to do is you have to get drug to the agent. So if you want to attack a particular cell, which has at the surface a certain protein, you better find a way to find that protein and dock to it. And then you attach an agent to your docking protein, and you're in business. Right? So the first thing, this is the simplest in vitro selection experiment. It goes back to Orff in the 60s, as far as I know. You have a tube of RNA. Okay? You have random RNA strands in the soup. You dip your sheet in there. You pull it out. And then the stuff that attaches to the surface, the protein, you wash it off. And then you multiply the washed off RNA back to the original amount. Everybody understand? Right? Something attaches to the surface. Something might randomly attach. Some things might attach because it actually really docks. You somehow wash that off. Yeah, this is all like, it's more like cooking, right? I remember I had a mad scientist once, Vichy was party once. Vichy was there. There's a woman there, she was a biologist, and she did extract DNA from onions. Really impressive. So it's all there. You just have to know how to extract it. There's all these techniques. But the kind of attitude she had when doing her experiment was, it looked to me like she was just kind of cooking with a little bit of enhanced techniques. Okay, so you can modify this experiment by adding a little bit of a similar, you want to attach to a certain protein alpha, add alpha prime into the soup, which is a slight modification, a relative of alpha. So that means only things that are very specific to this particular protein will attach to the thing. So you could make it more selective. There's all these tricks, right? You would come up with them if you played in that area. You put a similar protein into the solution. So the point is that this cannot be done with a computer. Why? And I'm not a physicist, but you supposedly have, this is Avogadro's something, 10 to the 20 RNA strands per liter of soup. 10 to the 20 is too big for computers. You cannot simulate them, and you cannot simulate them in particular in 3 dimensions. And as a matter of fact, the recent Turing Award was awarded to Les Valiant for, this is very good for our community, it gives our community visibility, for his research, amongst other things, in machine learning. He invented the PAC model. And Leslie Valiant postulated a model of evolution where essentially it's complexity theory based. It says, you have a bunch of variants around, and then you have a Turing machine that's allowed to spend polynomial time to modify the variants, mutations, and then there's another Turing machine that selects. But if you listen to his stuff, he cannot find any interesting application, really, of this model. It clearly, from a complexity theoretic point of view, all his steps incrementally are reasonable, yet he cannot explain why nature is so smart, why nature evolutes so fast, and why it does all these interesting things. My answer to this is complexity theory is important, but we cannot fathom how much computation happens in the small, kind of like what Feinstein was saying. There's a lot of room in the small, and this kind of computation with 10 to the 20 RNA strand, you cannot simulate in the computer. We're not there yet, and that's where the interesting thing happens. That's my suspicion, but I'm not sure. Okay, good. So what's the basic scheme? You start with a unit amount of RNA, you do a functional separation into good and bad RNA, and then you amplify the good ones, the one that you want, to unit amount. Okay? So now I have to explain to you how you multiply RNA. Actually, you take RNA, you convert it to DNA, and then you do a duplication process. And that was invented by Kerry Mullis, and supposedly it was invented while he was on an LSD trip. somewhere on some hot tub in Lake Tahoe, near Lake Tahoe, because there's these the natural hot springs. And he had to, he wanted to figure out how duplication of DNA happens. And he wanted to do it in the tube, right, in vitro. And so he somehow thought, well, this is a life form, an extreme life form, which happens around 95C. It survives there. It does DNA duplication. Let's try to figure out what enzymes it uses. He extracted the enzymes, and then based on that, he built a methodology for doing it in vitro. Okay? So this is one of the most technically, as far as technical advances in biology are concerned, this is a major one, right? Okay? So I'm going to show you a little something now, if it all works, since we multimedia thing. Okay, so click here, Apple F. I had a lot of help. Okay, so now we're going to explain to you how this duplication works. This is used, of course, in crime scenes, and this video is sort of geared towards explaining it to a policeman, I guess, how this duplication thing works. Okay, PCR works. So here's your tube, you see.
A sample of chromosomal DNA, also called genomic DNA, can be used as the starting material for the polymerase chain reaction, or PCR.
PCR.
With PCR, an investigator can amplify a single copy of a DNA segment into billions of identical copies. The DNA segment of interest, or target DNA, is indicated in red. In addition to the target DNA, a PCR reaction contains several other ingredients.
These guys fit at the ends, the primers.
DNA primers.
These are the alphabet, A, C, G, T, whatever.
The primers are typically about 20 nucleotides long and are complementary in sequence to the ends of the target DNA. Taq polymerase is derived from hot springs bacteria and can tolerate the
that's what he found.
Of a PCR reaction. A PCR reaction lasts several hours and typically consists.
Now it's seconds. Several hours, now seconds.
A cycle begins by heating the reaction mixture to 95 degrees Celsius.
Then it denatures.
The heat denatures the DNA, breaking the hydrogen bonds that hold the strands together. After denaturing the DNA, the temperature is reduced to around 60 degrees. So that the primers can form hydrogen bonds.
These are shorter, so they attach first. That's why they attach.
Note that the primers and the target DNA follow base pairing rules. An adenine, A, pairs with a thymine, T, and a cytosine, C, pairs with a guanine, G. In the next phase, the temperature is raised to 72 degrees Celsius. Taq polymerase functions optimally at this temperature and begins polymerization.
This stays attached, the primer stays attached. And now it zippers up, it's like a zipper.
After one complete cycle, there are two double-stranded copies of the target DNA.
Completed. It goes all the way to the end. Usually, in my application, I would only have these short pieces around in the first place, but this is a crime scene application.
It contains many copies of the primers and an abundant supply of nucleotides to perform many additional cycles. After a second cycle, there are 4 copies of the target DNA. After cycle 3 is finished, there are 8 copies of the double-stranded target DNA sequence. Note that only 2 of the double-stranded copies consist of just the target fragment. The others also include flanking DNA regions.
These grow linearly, the other ones exponentially.
As the number of cycles increases, the products consist of a greater proportion of fragments with just the target DNA.
Exponential and linear, you idiot.
After 4 cycles, half of the fragments consist of just target DNA, and half of the fragments also contain flanking DNA. With each additional cycle, the number of copies of our target sequence doubles. At the end of cycle 25, there are more than 33 million copies of this double-stranded target region.
Okay. So thanks. I just stole this off the web, you know, like a good computer scientist. So again, Kerry Mullis. So now DNA is extremely boring. It's an information molecule. The chemical and biological property of DNA is very similar no matter what information is loaded into it. RNA, it has 4 letters. Nature got it wrong. It has 4 letters instead of 2. RNA is 4 letters as well. It's a little bit more reactive. Some functional groups, the sugars are changed a little bit. And, and it's a little bit more reactive. It folds. And some of the early enzymes, supposedly, that generated life are based on RNA. Now, once you have RNA, there's another translation via a very complicated enzyme, or whatever you want to call it. It's very, very complicated. Studied by many people, Hernoller, for instance. It's turned into a protein, and that's alpha beta size 20. Now, proteins is amazing. Proteins are amazing. 20 alphabet, you can have amino acids, the elementary things that are lipophile, they like water, they have a charge. You can basically build all the materials that you see here using these proteins. Okay? So it's the building block for life. Ideally, you want to do a selection based on that. And people have played around with this, and supposedly it's working. So there's a trick. You see, this is a one-way direction, so you cannot select and translate back. So what people do, this is something a computer scientist would immediately have invented. Unfortunately, you don't see it in the screen. Yes, so it's up in here. You don't see it. There's the DNA molecule attached here to the protein. It sits up here. You don't see it. So you select based on the protein, and you tag them with the information, with its encryption. You understand? So it's attached. So now, so what you need to do is you initialize the tube with tagged random protein stands. The tag of a protein is the RNA encoding of its protein. Then you do the functional separation. You retrieve the good protein and the tags from the good protein, and then reproduce protein from the retrieved tags with a ribosome. Okay? So this means you have to duplicate now tagged proteins. So you have the inscription, you build the protein, and then you have to have a mechanism at the end attach the DNA that you just read to the end. And this is a technical problem, and people are working on it. Okay? So if you have that, you can do functional separation with proteins. Okay, so this is technically very difficult, but supposedly almost doable. The problem with this kind of a thing is, in general, is there is not many, and there's interesting specific functional separations that you can do. You know, you can do attach, right? You can do stuff like that, but you cannot do, does it clean the diaper? Which is a more complex thing. So pour something into the diaper and then retrieve the ones that cleaned well, that's messy. Okay? So in some sense, this thing doesn't work that well. Of course, some people are still doing it a lot. But I was interested in this from an information theoretic, from a computational point of view. Okay? So this is biology. We want to do math. Now I'll show you the transition to math and to neural networks. Totally cool. So number the unknown RNA strands from 1 to 10 to the 20. Just give them names. I don't even know the names. Right? They're just some names. Assume they exist. Okay, now, WI is the fraction of RNAi. WI is clearly, it's a concentration. It's clearly bigger than 0. Yeah? Good. Now, what's the contents of the tube? The contents of the tube is a vector. It's a vector of concentrations, right? This is how much you have of the first, the second, of the nth. N is 2 to the 20, 10 to the 20. Okay, it's a concentration vector. This is a probability vector. So the content of my tube is a probability vector. So this is what we usually work with, machine learning now. Right? It's a probability vector. Good. Now, the sheet of protein, yeah, it sums to 1. The sheet of protein, I'm going to now describe that as another vector. The sheet of protein, xi, this says it's the fraction of 1 unit of RNA that is good. The fraction of 1 unit RNA that would attach to the sheet. The percentage of attachment. Okay, that's another number. A number between 0 and 1, xi. Okay, again, the protein is represented by a vector from 1 to 10 to the 20. Now, the beautiful thing is that the vector that's my tube and the vector that's my sheet, those are unknown. It's blind computation. It's like kernel computation, right? You don't really look at the individual features. You work with it, but it's unknown. I don't know how to formalize this. I called it blind computation. I just like it. Okay? It's blind computation. You don't know what it is, but you just still can work with it, right? Now, of course, every good talk has to have a deep assumption that is totally unrealistic, and here it is. The fraction xi, I assume, are independent of the concentrations. How much attach is independent of the concentrations. At low concentration, this might be the case, but probably it's unrealistic. Okay? So, but that's in what I do, that's what I assume. Okay, so look here. I showed you the two vectors, right? The tube and the sheet. Now here, what's the good RNA? The good RNA is, well, how many of the first one attach, plus how many of the second one attach, plus how many of the nth one attach? Oh, dot product. Voila, neural networks. Neural networks. So when I do this, I dip it in there, and I wash off, I have computed the dot product. This is the bad RNA, the complement. What's the amplification? Well, the good RNA is multiplied by a factor. Everybody is assumed to be multiplied equally as much. I'm going to talk about that in a moment. If precise, then all good RNA are multiplied by the same factor. So this is multiplied by w dot x so that the total sums to 1. Well, if the total sums to 1, then the factor has to be 1 over that. It's just trivial. Okay, so this is the update. So, voila, what did I do so far? I tip this in, I wash it off, I amplify to unit amount, and what I have computed is this thing, this update. Now, what is this update? Anybody see what this is? It's an interesting update. I claim it's Bayes rule. Wi is the prior, xi is the probability of good given I, w dot x is the probability of good. This update is Bayes rule with 10 to the 20 variables. Forget about computers, you know. You just do it in your kitchen. Yes?
How do you do gift sampling this way?
I don't know. Maybe you could. I don't know. Okay. So now, here's I have a lot of fun with this, with these plots. So, so what I did here is, this is now the content of a tube at time 0. I have a tiny amount of the red agent. I have 3 RNA strands. A tiny amount of the, reasonable amount, 0.4 of the blue, and about 0.6 of the green. You can also view this as species, as concentration of species, and then the survival factors are these, and the species are evolving. So you could view it that way too. So at trial 10, these are the concentrations. And I assume that the update factors, the survival factors, of the red one was 0.9, rather high. of the blue one was 0.75, and this one was 0.8. Okay? So here's what's going on. I do the same update iteratively, okay, to make a point. I had a lot of fun figuring this out. So look here. So 7.5 is less than this factor. So initially, they don't know about this guy. So this beats the blue one. It goes up faster, and this one goes down. It tanks. But then the red one comes out of the dark and beats them both. Yeah? So I'm going to do this kind of experiment again with more. I have 4 agents now. 0.9, very little difference between these two. This is slightly bigger than this one. Initially, it beats it because of the little difference. Multiplicative update, any big little difference makes a difference. This one tanks, but then this comes out of the dark and boom, goes up. Now, if you write the update, it's basically this. WTI is W0i xi to the t, because you did it t-fold. T is a speed parameter or a learning rate. In Bayes, they always have learning rate 1. I like to do Bayes with learning rates different than 1. 1, 1.2, 2, you know, much more interesting. You can still do Bayes, and you can do a whole calculus based on that. So this is this kind of evolutionary thing. The trick is to see it go backwards in time, too. You know, you always want to cheat. So here you go forwards in time. If I run this guy backward, its survival factor is 1 over 0.54. You see, the survival factor become the inverses. And now you have the situation, you run it with 1 time step forward, and so 1 plus minus 1 is 0. So 1 time step forward and backward gets you where you were. Okay, very cool. And then 2 half steps get you 1. Okay, anyway, so you get these curves, and there's 2 sigmoids in there. This one, the one with the highest, and then the one with the lowest. And the other ones are sort of attached sigmoids. So this is kind of the growth curve you get. Blessing and curse of the multiplicative update, one guy wins. One guy wins, and that's the problem. Oh, I have this fancy thing. Okay. So best are too good. Multiplicative updates, factor. Best get amplified exponentially fast. Best wipe out others. Loss of variety. If you want variety, multiplicative updates are very bad. Okay. Can here come up, I have a paradox. So imagine you have a tube. Right? I did it in a square. So you're looking at the tube from the top. Something like that. Okay, you have a square. And then in that tube, you have 15% A, B, 60% B, 25% C. And it's mixed together. And you don't even know what ABC is. I want to make more of the soup. How can I do it? I want to make more of this soup. There's information in the DNA, in the strands. I want to duplicate it. But I don't want to sequence the individual guys. I cannot go in there and fish and look at it. Blind computation. How do I do it? Anybody knows? This is a very deep paradox. So, because if you just apply PCR, then it happens to be that maybe A is shorter than the others, and it gets duplicated faster, or it somehow hugs the Taq polymerase in a certain way that favors the speed. It has maybe certain letters get duplicated faster, and Taq has more, or there's more stuff in the soup. Anyway, the factor is not perfectly, never 2. There's always a slightly different factor for the different letters. And if you duplicate this with independent duplication with PCR, if you do it long enough, because of the multiplicative nature of your update, one guy will win. One guy will win. You cannot do this just with PCR. You have to do something else.
So does you mean that if you do PCR, B will dominate?
One will dominate, yes. One.
Not necessarily.
Who knows which one it is? You don't know what's in there. But if you do PCR for a very long time, one guy takes over. They see that in vitro selection all the time. They do some experiments to do a lot of PCR. One bug that is not interesting to anybody, bug means DNA strand, will dominate the whole soup. You have to throw it away and start new. It gets contaminated. Yes?
So just as a side question, do they have to sometimes be very careful how they clean whatever they're trying to get the DNA off of, so that there's not a chance that it can
Say it again, how they?
Like, do they sometimes have
so they don't accidentally introduce
Yes.
next down.
Yes. Right. You have to clean your sample so you don't get any stuff that you don't want. So to do this in the soup, what you need to do is the following. You take your strands, and then what you do is you can have enzymes that add little strips to the end of your RNA strands. So now, this is a strand, and at the beginning and end, there's certain extensions that are put on. Extensions here and extensions here. Okay? Then you heat it up and let it put together, and it zips together in long strands. So after that, you have long strands. Oh, I have this. And you have long strands of A, B, 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 something like this, and they're put together with a known sequence here. Okay? So now what you do is you take this long strand, and you fish out one, or a small number, and you can do that using very simple tricks. You amplify the same long strand many times with PCR by adding primers to the beginning and end, so making sure that the whole strand gets duplicated. So now, when you want the soup, you chop in the constituents. So this becomes your encryption of your soup. It is true that the concentrations on this long strand are not going to be perfect. They're not going to be perfect. They're not going to be quite 15%, 60%, and 25%. However, they're going to be roughly right, and they do not blow up. They're coupled. So it doesn't wander off. You prevent the curse. And this is exactly why we have chromosomes in the kernels of our cells. We have chromosomes there because if you would have the genes float out freely, who would keep track of the mess? They would compete with each other. Some self-if genomes would survive, dominate the soup, and the variety that is needed to get you into the next generation wouldn't be preserved. So there's mechanisms that have this very long chain. For some reason, it's not just one. It's a small set of chromosomes. And during meiosis, it's made sure that the full set is duplicated. The full set. Totally cool. Right? And nobody's cheating. And if there's the wrong number, you have an additional number of chromosomes, it's a big problem. There's Down syndrome, etc. So free-floating genes in the nucleus would compete, and you would have a loss of variety. Everybody get the point? You have to couple them. You have to couple them. Okay, so now here's pictorially what's happening. If you duplicate with PCR, if A and B is free-flowing, then they compete, and one will win. Let's say A. Competition, one will multiplicative update. There should be a loop here, right? If they, however, hooked together here and go into this thing together, then A and B should cooperate. They should cooperate and to get more of A and B across, because then they will win together, cooperation into the next generation. Breaking the curse of the multiplicative update. Very deep thing. Now this multiplicative update versus how to prevent it happens at all levels. I'm going to do it, I do it at the molecular level. Later on I will do it at the species level. It's a very, very deep philosophical problem. Okay? Good. So this thing here, this connection is like the handshake. We are going into the next generation together. And that's actually what, if you read Dawkins, The Selfish Gene, he talks about that. Genes cooperate, and a genome is a cooperative arrangement to go into the next generation together, and we, the bodies, are only there to propel them into the next generation. It's a pretty roundabout way to duplicate yourself. First, you make these guys have a baby, and then squeeze out the baby and do all these things, and then go through school so they make more babies. That whole thing is an orchestration by the genome to duplicate themselves. Right? It's not funny. I'm teaching you the basics. What?
together, then you have them cooperate together, and they move on, you get PCR, and then they're all together. If you didn't know what they were in the first place, how do you separate them when you get them all together at the end?
Okay, this thing here, you had a particular glue here that you knew. You put an enzyme, you can chop it up again.
Okay.
And actually, you're going to keep this around as your template for your soup. It's a signature, right? Totally cool. Look, it's just information theoretically. You have to do it that way. There's no other way. It's totally cool. OK. So what I'm asking myself is, what updates represent an iteration of in vitro selection? What updates are possible with blind computation? You must maintain non-negative weights. There's slight motivation. You could do good and bad and modify the factor a little bit. So far, we did this. The question is, what else can we do that is sort of reasonable and feasible, and it updates concentrations? And now I'm going to show you, I'm going to lead you to a technique that we invented in machine learning, and it goes back to the Winnow algorithm. So imagine for a moment, you have your sheets. They're different proteins. Now they have 5 different ones. And you have your little selection procedure here based on attaching to the second, to the third, to the fourth, to the fifth. You do it cycle over them, all 5 of them. So in trial T, you do functional separation based on xt. So far, all the T were the same. But now I want to attach to all of them. Do you see? Not just one. I want to learn a union. And I want to learn it in a decoupled way. And that's what Winnow does. Totally cool. This is why Winnow has something important to say about in vitro selection. It's a commonsensical thing, but it's totally interesting. The feedback between the two, that's what I'm fascinated by. So imagine for a moment there is 2 RNA strands that can do the job. 2, I called them 1 and 2, but they could be 50,376 and some other number. Okay, it doesn't matter. I just called them 1 and 2. The first one has this attachment factors on P1 and P3. It has high numbers for these 3 protein sheets. And the second one has high number for these 3. There's some overlap. You don't know which ones they are. It's blind computation. You know there's 2 in your bowl. OK? So there's 2 around. 2 cooperatively solve the problems. The goal is you want to arrive at this tube. You want to bring those 2 up. How can you do it? Well, the problem is, if you want to do blind computation, and you overtrain with the first 2 cases, then you bring up the first guy and it wipes out everybody else, and the second guy might not even be in your soup anymore, so you're lost. Curse of the multiplicative update. If you overtrain with the other one, and if you have a huge setup, you never know who's going to win, and you want to do blind computation. So how are you going to do this? The xt and the w are unknown. Any idea? Interesting. So what you do is you need to have some kind of feedback in each trial, and it has to be reasonable. And first I give the related problem in machine learning, where the tube is a weight vector, the proteins are the instances, and you want to learn a disjunction. And you basically want to do something like w.xt is bigger than a threshold. You want to learn a disjunction. That's exactly the Winnow problem. And the way Winnow does it is the following. It measures whether you are bigger than a certain threshold. When you do your functional separation into good and bad RNA, you look at the good RNA, that's this one, and you measure, is there more than a certain amount? If there's more than a certain amount, then you don't update. You just keep the soup that you had. Only when your concentration is too low, when this goes the other way, then you multiply your concentrations by a fixed factor and renormalize. We call this a conservative update. This is the same thing as the perceptron. In the perceptron, when you're on the right side, you don't update. When you're on the wrong side of the threshold, you update. Since you're now in perceptron, you do additive update. In Winnow, you do it in the exponents, so they become update factors. Update factor of 0 corresponds to factor 1, stay where you are. And then if you're on the wrong side, you update. There's a paper called Winnow versus the perceptron. You can look at this. Okay, so you everybody understand? You make the update conservative. If you're already if you already noticed, if you already attach a lot for this thing, you don't update based on it. You don't do your PCR in this whole little loop. You just recover everything. So this is makes this update conservative. Now, there's beautiful things that you can prove about this update. You can show that it's information theoretically optimal. The number of mistakes, the number of wrong updates it makes, or the number of updates it needs, essentially, is k log n over k. Beautiful because n is huge, 10 to the 20, but there's a log there, so it's still reasonable. That's why the multiplicative updates are so interesting. They are log, the generalization is log in the number of features. Here, this is very important. You cannot do an additive update. Why? Because usually they don't learn until they have seen dimension many examples. But dimension is 10 to the 20. But log of n is fine. So the fix was when the good side is large enough, then you don't update. You do a conservative update. Okay? So you can simulate normalized winnow. You can simulate this kind of update at the molecular level using in vitro selection. Okay? So I showed you 2 tricks now so far. And we're going to learn a number of tricks today. 2 tricks. Usually you came late. So OK. So I waited. So now, 2 tricks, the handshake trick, and here the conservative trick. OK. OK, now I'm going to do another trick. And this time I'm going to start in nature again, go back and forth. This is the super predator idea. It's very important. I teach classes with evolutionary biologists and game theorists, economists. And here's the idea of a super predator. If you are a lion pride, then what you do is you go on the bluff and do a histogram of all the different species that are around this year, and then you decide this is the most frequent one, so you specialize on that. Of course, this is not true. The lions are not doing anything. All the work is done by the lionesses. They're the better hunters because they don't have this big scruff around that's in the way. But nevertheless, a lion pride will sort of automatically specialize on the most frequent species around. It has to learn how to hunt a species. Like buffaloes, you have to attack from the back. It has to be learned. And there's experience. And different prides have hunting different things. So they specialize. There's an investment. Since there's an investment in learning how to hunt a species, you better invest into the most frequently occurring species. That corresponds to capping the concentrations. And capping, we know from machine learning, right? SVMs, we use capping. And that prevents the multiplicative update. We discovered that as well in machine learning, as you will see. So we do a multiplicative update, but we cap. So if you have a multiplicative update, you run into the corner of the simplex. Well, if you cap, then you run into this capped simplex, and it has more corners. They correspond to sets of size 2, as you will see. How do you cap? If you're out here and you want to project back in here based on relative entropy projection, you take the weight that is above the capping value and you distribute it in a multiplicative way amongst the remaining guys. So you're learning now sets. You prevent the curse of the multiplicative update by learning multiple goals. Not a single goal, but here sets of size 2. Here again sets of size 2. This is the 4-dimensional probability simplex, which becomes a tetrahedron. I didn't bring my props. So the combined update converges to the best corner of the capped simplex, which learns sets of size m. So capping lets us learn multiple goals. So how to cap in the multiplicative in the in vitro selection? So in the in vitro selection setting, how would you cap? Well, you could maybe do sloppy PCR that says frequently occurring things will attach to each other and block each other, and therefore they're in the way of their own multiplication. I don't know how to do that, but that would be a thing to do. It's a problem. in general, when you do in vitro selection, it can always happen that one guy dominates everything and your soup is useless. Okay? Good. Now, I want to do another trick from machine learning, and it also kind of inspires something it's also something that's done in biology. Okay, so I have experts. Yeah, I want to predict the timeout on a laptop, right? So what I do is I invent about 20 different experts, which are different timeouts. I run a multiplicative update on top, and then you average and predict with that. So here's the different timeouts, 20 of them or 100 of them, whatever you want. You maintain a weight. The masked algorithm maintains a weight for each of these timeouts. And then you update the weights. This is a factor between 0 and 1 of the energy used by the timeout. So the ones that use a lot of energy go down multiplicatively, yeah, over normalization. So this is sort of a nice trick. The key thing is that if you do this, you have this bursty behavior of the usage time. These are the different idle times, the different lengths. Okay, if it's very bursty, this favors long timeouts. You stay up, and this one favors short timeouts. So you need different timeouts for the morning, for the midday, for the holiday season. You need different timeouts, different typical lengths, and you have to adjust automatically. If you do a multiplicative update, you're screwed, because you do a multiplicative update during this regime, one update time is favored, all the other weights are wiped out, and then you cannot recover. How do you fix this? In machine learning, what we did is we do a multiplicative update, but then we take, let's say alpha is 1%, 99% the weight we just got, and 1% we mix in the uniform distribution. Okay? So, and in biology, they do that as well. When they keep a beaker, a huge trash can of their initial soup, and when their solution gets saturated and favored by one agent, then they just mix in a little bit of the initial. That's what this update is. We call it sharing to uniform past. I did a lot of work on this with Mark Herbster. Now, this facilitates switch to this facilitates a switch. Now, if you want to do something really fancy, you keep track of the average weight of the past, and you mix in a little bit of the past average. And this is a totally fascinating update. It's a talk in itself. It's a multiplicative update combined with mixing in a little bit of the average. The multiplicative update is the short-term memory, and the average introduces a long-term memory. So you switch very well to something that was good in the past. It facilitates a switch to previously useful vector. It's a long-term memory. It's my theory of how the brain works. But I can talk about that over beer tonight. OK. So the question is, how is the long-term memory implemented in nature? And I believe it's one of those things. It's there as well, because the curse of the multiplicative update is there. So we have all these funny things that happen. Sex, we have junk DNA, and I think they all ameliorate the multiplicative update. So far, I showed you conservative updates, learn multiple goals, upper bounding, learn multiple goals, lower bounding weights, robust against change. So I'm basically done. What I did here, which I have done before, in the slides I showed where these multiplicative updates come from. Additive updates, I did it in linear regression. I did these slides already. Additive update looks like this. Multiplicative update looks like this. This you cannot do for updating concentrations. This one you could implement with concentrations. It's called the exponentiated gradient algorithm. It's motivated by a trade-off. And here's relative entropy trade-off gets you the multiplicative update. You need Lagrangian. You can do it with differential equations. I did this. I added slides to my boosting talk, lecture 2, I think, where I showed you also the implicit, the forward and the backward Euler. You can look at those. I don't make that point here. Here's the multiplicative differential equation. Summary. They converge quickly, but wipe out diversity. Converge quickly, but wipe out diversity. Changing conditions require reuse of previously learned alternatives. You want variety. Variety is important. It's a requirement for success. These multiplicative updates occur in nature. They have to be ameliorated. What do you have in nature? At the molecular level, you have chromosomes. I talked about that. Then you have super predator setups. Sicknesses is another kind of a setup. It prevents, it caps things. If you're too concentrated, being together with your kind creates a vector and the sickness travels more. Therefore, the sickness spreads and wipes out, so everybody becomes isolated little groups. So it's a capping phenomenon. Sicknesses is capping. Very important. Very important. If you don't have that, you have the curse of the multiplicative update. Another one is mutations, linking, super predator, sicknesses. The machine learning, we had the conservative update, we had capping, we had lower bounding the weight, weighted mixtures of the past, all these tricks. And they sort of, in flavor, similar to what you would do in vitro selection. And it was an interesting exercise for me to notice the connections. Okay, one more slide. Well, there's a version of the multiplicative update that I'm very much fascinated by, which instead of having a probability vector, a concentration of species vector, you have a density matrix. What is a density matrix? It's a probability vector with an eigensystem. Symmetric positive definite matrix of trace 1. The update goes like this. The new weight is the old weight, matrix log, minus the gradient, which is another matrix. You subtract them, or add them, whatever you call it. Matrix exponentiate, divide by the trace. This is beautiful, beautiful math. Before, the multiplicative updates were motivated by relative entropy regularization. Now it's quantum relative entropy as a regularizer. I don't even know whether something like this is implementable in nature. So I'm kind of curious by that. Maybe it is. I don't know. I don't know. So that's what I'm fascinated by. It's not quantum physics, because in Schrödinger equation, you keep the eigenvalues fixed. And here I operate a lot on the eigenvalues. It retains the initial case. The diagonal case is a special case when all these matrices are diagonal. Then you get the normal multiplicative updates. There's a generalization of Bayes' rule, totally beautiful, that goes with this. You know, prior density matrix, take matrix log of data likelihood matrix, add them up, exponentiate, and that's your Bayes' rule. Totally cool. It all works out mathematically. Not quite sure what it means, but it's awfully pretty. And I'm looking for a physical interpretation of it, which I don't know. And I'm out of time. Any questions? You can do anything. Anything about life. You know, it's my last lecture, so you can do anything. You can ask anything. Yes?
Can you give a concrete example of mixing this 1 over at this uniform distribution back in a natural setting?
The natural setting in nature?
Yeah.
In nature, the uniform distribution might be mutation rate. So you always go back to the base mutation rate. It's one of those things. So you cannot do the multiplicative update all the way perfectly and home to one particular guy, right? There's a mutation rate. So it's really the philosophy that comes out of this. Darwin stressed the selective process of evolution. The selective process of evolution is the multiplicative update. What I'm doing here is I am stressing the curse of the multiplicative update. So short term, Darwin's philosophy and the quick understanding of evolution is it's mutation and selection. But long term, not it. Because imagine for a moment, you are so good with a multiplicative update and you do it at high speed, one particular guy wins. Very bad, very, very bad against change. You also have long-term thing. So there's a tension, you see? And that tension between short-term and long-term, which we talk about in machine learning all the time, that's that learning rate in some sense. That happens in nature. Nature has to tune that too. It has to tune how fast you should mutate, how fast you should evolve, how fast you should have offspring. If you do it too fast, you have too much curse of the multiplicative update. And then only those systems survive that in the long term have some kind of mechanism to ameliorate the curse via these unlucky things that we're trying to get rid of, sicknesses. They have a function. Yeah? Mutations, they have a function. Predators, they have a function. They have the function to prevent the curse of the multiplicative update. That's what I claim. So it's, yeah, it's interesting.
Since we are running out of
Oh, yes. OK, thank you.
Okay, so